What I was asked was to write a certain number of questions, and that's what I'm going to be doing. But I'm not 100% sure, to be honest. Wish I could give you a definitive answer. Okay, if you don't mind, I'm going to try to get going just a little bit early. I think we have about 1 minute before we officially have to get started. I've been asked this question several times today, and I have no definitive answer for you. I only know that I was asked by Dr. Key to make sure that I supplied 26 questions, so I don't know if the endocrine section is going to be on your term test. I'll ask him to post an announcement about that later on this evening. In the worst case scenario, I'm assuming that it would be somewhere around 3 questions at maximum from the endocrine section. Um, and again, I'll post some more about that tomorrow during the review session about the specific breakdown of the number of questions that I was asked to provide for each of the different lectures. So for today, for example, for today, I was asked to provide 6 questions from all of the lecture material that I'm going to be giving to you, and that's exactly what I've done. Okay, so we're going to be talking in sort of a full circle. We're going back to talk about muscle. So we've been looking at sensory receptor systems. We've been looking at how these reflexes work. We've been talking about muscles, but we actually haven't looked at the mechanism by which muscles actually do their job. So we're going to go back and talk about muscle. And in particular, we're going to be looking at striated or skeletal muscle, and that's voluntary. You control what's going on. Do you have any idea what the benefit of having striated muscle is? So controlling movement, but think more philosophically. This is our last lecture together, so think a little philosophically. Why bother with striated muscle? Like, what does it really allow you to do? It allows you to move, absolutely. It allows you to do all sorts of things. But philosophically, why would we want to actually look at skeletal muscle? It's really deep. It's really deep. Honestly, it is. I read this as a response that a student gave me one time, and I actually thought it was really deep. It is your only opportunity. All your other muscle systems, whether it's smooth or whether it's cardiac or skeletal, they're all involved in keeping you alive. But the skeletal muscle system, the one that we're going to be starting off with, is the only muscle system that actually allows you to leave a legacy behind. So whether you're leaving behind a note that you've written with your skeletal muscle, or whether you're saying something, or whether you're typing something out, only your skeletal muscle system allows you to leave a lasting legacy. Nothing else will allow you to do that. And it's maybe one of those evolutionary traits that skeletal muscle allows us to actually make use of. So this is what we're going to be talking about. There are going to be some definitions that we have along the way, and I will point out things that you should make sure that you go and spend some time on. So we're going to start off with the motor unit. I'm going to give you a definition of the motor unit. Then we're going to be talking about different muscle types. All of these are related to skeletal muscle. All are these skeletal muscle types. Then we're going to be looking at the mechanism by which skeletal muscle actually contracts and why we need a nerve impulse to cause skeletal muscles to contract. And then we'll be talking about some of the operational physiological definitions for how muscle contraction actually occurs. And this gets into the basic property of the muscle fiber itself, which is called the sarcomere. So this is the motor unit, and the motor unit is the motor neuron. So it could be like one motor neuron and all of the muscle fibers that it innervates. So this is a muscle group that you're taking a look at here in this diagram. And you can see that, for example, this sort of pale pink or orangish motor neuron in the ventral horn is only going to be innervating a certain number of muscle groups. None of the other ones, only specific ones. That would be one motor unit. We might also be having another motor neuron very nearby that will go and innervate these darker fibers, the darker pink fibers that you see here. And so this dark pink motor neuron and its associated muscle fibers that you see here represent another motor unit. And we can do the same thing with this motor neuron here and all of its individual muscle fibers. And you can see that every motor neuron has its own group of muscle fibers that it is innervating. All of these together, when they're contracting together, will cause the entire muscle to contract. But you can see that the entire muscle actually consists of different muscle fiber types. And again, we're going to talk about these different muscle fiber types in a few moments. So all of the muscle fibers in that motor unit, when this motor neuron is activated and it releases neurotransmitter, it is going to cause all of those specific muscle fibers to contract together at the same time, which is what is being relayed to you in this second bullet point. So in order for you to have really smooth muscle contractions, the types of muscle contractions, for example, that you have in a reflex activity where your muscle spindle has been stretched, you need to be able to activate those muscle fiber groups, these individual groups, all at the same time. And the number and the timing of these motor units will tell you like how much force and power you generate, how smooth the activity is, etc. One really important point that I'm going to emphasize several times here today, muscle contraction always begins with the smallest motor units being activated first. And it's very important that you understand this principle. And this is known as the size principle. So this is where we're going to see it first, about these small motor units being activated. And these small units are activated first because they're the easiest to excite. So we start with the smallest, easiest to excite, and then we recruit bigger and bigger motor units because they're harder to actually excite. So what are some of these different muscle fibers or motor unit groups? So here's like one type, and there's another one in a different color, and then there's this third type. So these muscle fibers in a motor unit are all of the same type. In one motor unit, we only have fast glycolytic being stimulated by that motor neuron, or slow oxidative being stimulated by that one motor neuron. We're always talking about the same muscle types or muscle fiber types, but we can actually categorize the different muscle fiber types into these three broad categories that I'm going to be mentioning to you. We will be looking at the intermediate type, the slow oxidative type, and the fast glycolytic. And those are the three muscle fiber types that I want you to make sure that you know. And one of the ways that we identify these are through the use of different histochemical techniques. So for example, if you were looking at fibers that are capable of responding both quickly as well as slowly, the muscle fibers would be enriched for the myofibrillar ATPase, and that is unique to those muscle fiber types. If we're looking at muscles that are really good at oxidative types of reactions, then we will look and stain for succinate dehydrogenase. We would see that in more of this type of muscle. And if we're looking for muscles that use a lot of glycogen stores and have and use glycolytic potential, the third type, this would be represented of the fast glycolytic muscle fibers. We would go and stain the muscle fibers for this alpha glycerophosphate dehydrogenase, or GAPDH. So when we look at the different characteristic features of these slow oxidative types, so you might go back and you might think that we might be staining it for succinate dehydrogenase. These slow oxidative muscle fiber types are found in different muscle groups, and these are identified as being slow twitch and very slowly contracting. So they have a very, very long rise time before you can generate sufficient force. One of the things that defines this, in addition to succinate dehydrogenase, for example, is that in this particular type of slow oxidative muscle fibers, these tend to be rich in the protein known as myoglobin. And because myoglobin is a pigmented molecule, the muscles that we find myoglobin in, the slow oxidative muscle fiber types, tend to be a dark red color. This is also true because they have a lot of blood vessels going through, and also because of the myoglobin. In general, slow oxidative fibers are darker in color and dark red in color. So when you're talking about, for example, when you talk about looking at different muscle groups that you're eating, so if you're eating chicken and you're eating dark meat in the chicken, you're eating dark meat in the chicken because it has lots of myoglobin associated with it. And this type of muscle fiber group is this slow oxidative type. It also has many different mitochondria within this muscle fiber, and so as a result, we know that it involves this oxidative metabolism. What makes this kind of interesting is, with all of the biochemical background that this provides, so having myoglobin, having lots of mitochondria, having succinate dehydrogenase, using oxidative oxidation as its power source, this represents a non-fatiguing type of muscle. It doesn't generate a lot of force, and as a result, because it doesn't generate a lot of force, you can actually contract this muscle for long periods of time. So these would be the muscles that might be involved in low-level force production that allow you to stand upright for long periods of time. It might be in the soleus muscle in your lower leg, for example. It might be a muscle that's involved in maintaining your posture. You can do that for long periods of time because it uses oxidation. It is a low force generating. It's not like your biceps where you're generating lots of power or tension. You can do this for longer periods of time. This is different than the fast oxidative / glycolytic. And again, this goes back to being able to identify both fast and slow fibers. So this would contain that myofibrillar ATPase. And this sort of has an intermediate twitch rise time. And I'll show you this diagrammatically in the next few slides. This generates sort of moderate levels of force. So they're good for a certain amount of power, but they're not the most powerful muscles that you have. Because it's sort of an intermediate type of muscle, it's oxidative and glycolytic. There's a little bit of myoglobin, so it tends to be somewhat reddish, not quite as dark red as the oxidative type, but it can also use anaerobic pathways, so it can also use glycolysis. So during muscle contraction, when you're contracting your muscle, one of the things that happens is the blood vessels to the muscles as you're contracting them actually get compressed. There's less blood flow. And so you might actually need glycolysis where no oxygen is being delivered to cause muscle contraction to occur. As a result, because it's intermediate, we have a moderate number of mitochondria, not as much as we have with oxidative, not as few as we have with glycolytic, just sort of in the middle. It fatigues slowly, so it fatigues over several minutes, and this is the type of muscle group that you might use when you have prolonged anaerobic activity where you need relatively high force or muscle tension being involved. So if you're racing like 400 meters or 800 meters, these would be the muscle groups that you would use or need a lot of in order to do really well. The last muscle group, the fast glycolytic, it has a very fast twitch rise time. And again, I'll show you all of these in the next diagram. So all of these points about what twitch rise time represents. It also generates a lot of force. These are really powerful muscle groups. And in fact, they also have very large fiber sizes in general. There is almost little or no myoglobin associated with these types of muscle fibers, the fast glycolytic. And these muscle fibers tend to be very pale in color. So if you're talking about white meat, this is the white meat that you will have. Very few mitochondria. It's very heavily relying on glycolysis. And as a result, because you are generating lots of force, you're going to be going through your ATP stores really quickly. It's going to fatigue very rapidly, about a minute to 2 minutes. And a lot of the fatigue occurs because of lactic acid accumulation in these different types of muscles. And these are the types of muscles where you are generating power. So when you're looking at sprinters, if you're looking at high jumpers, if you're looking at weightlifters, those large muscle groups that they have and they're developing, they're the ones that will have fast glycolytic fibers in abundance. You'll have more of these in a muscle group than the other two types. So this is all about fast rise times as well as the amount of tension that is produced. So the fast glycolytic, in terms of power or tetanic tension, the amount of force that can be generated, the fast glycolytic is by far the most powerful muscle group that you have. It generates this power relatively quickly, but it also fatigues relatively quickly. We don't have a lot of time to be able to use this muscle. The intermediate type, the fast oxidative / glycolytic, is sort of in between, exactly as the name suggests. It has a relatively fast rise time, but it also fatigues somewhere in the middle. The slow oxidative is not really great for producing lots of tension or lots of power with this muscle group, but it also can maintain that same tension, that same level that it produces for very, very long periods of time. And again, those postural muscle groups are the ones that you should be thinking about with this slow oxidative type. So if you're talking about these motor units and the muscle fibers, they're not all being innervated by the same motor neuron, so that's really important. One motor neuron will only innervate one certain type. It'll only innervate slow oxidative or fast glycolytic or the intermediate type. You never have one motor neuron innervating all three types. Okay, so it's always one motor neuron for one specific muscle type. And of course, muscles in general, when we're talking about like your bicep muscle will generally have a lot of the fast glycolytic, but it will also have some fibers that contain the intermediate type, and will have a small number of the oxidative type. So we have all of these in abundance. So this is a very interesting and very controversial question. Can you actually convert your muscle fiber types? And again, it seems to vary a lot, and the current thinking is that a lot of it is really genetic. You can't really convert, for example, the slow oxidative into the fast glycolytic. You have to have a certain genetic predisposition. So if you watch the Olympics, for example, oftentimes an Olympic athlete has their parents be excelling in a particular sport or being really athletic in that sport to begin with. So in order to win a marathon, you would have to already have a genetic predisposition of having a lot of slow twitch fibers that are in the quadricep muscles to allow you to run long distances. And if you're a sprinter and you want to be winning the 100-meter race, then again, you might have to have the genetic predisposition of having a lot of these fast twitch, the fast glycolytic muscle fibers already. And training just helps you to develop it. You aren't converting any of the slow or intermediate into the fast glycolytic. Again, very controversial. Some scientists don't believe that it's not impossible to switch. You can, with training, maybe convert some of those muscle fiber types. But based on what I've been telling you about the neurophysiology, you're born with a certain motor unit, which is the motor neuron and all of the muscle groups that it innervates. It would be really hard for you to imagine that that motor neuron would strip itself from one muscle fiber and then attach itself to another one. So again, some of the physiology is relevant, and it kind of makes sense in that particular framework. So we're looking at these alpha motor neurons. Do you remember what the other type of motor neurons were that we were looking at? So alpha motor neurons, all of the things that I've just been mentioning, all of the different motor neurons that you see here, all of these here in the ventral horn are all alpha motor neurons. What was the other motor neuron that we talked about? Yes. So the gamma motor neuron, and I'll come back to that again a little bit later on. And all alpha motor neurons innervate what are known as the extrafusal muscle fibers. So I've just shown you three different types of extrafusal muscle fibers. Remember, the gamma motor neurons innervate that muscle spindle, which is the intrafusal muscle fibers that allow you to determine if the muscle stretch, the spindle reflex, should be turned on and resensitized or not. So small diameter motor neurons, and again, this is important. You should maybe circle it, maybe understand that this is related to the size principle about different motor units and different groups being activated. Small diameter motor neurons innervate those slow oxidative fibers. Intermediate sized motor neurons, ones that are slightly larger in diameter, axonal diameter, innervate fast oxidative / glycolytic, the intermediate type. And then the large axonal diameter motor neurons, they will innervate the fast glycolytic fibers. Is that okay for everyone? Yes, no, maybe? Okay, you need to make sure that you understand it, and you need to make sure that the smallest diameter are slow oxidative, the largest diameter are the fast glycolytic. I also told you a few slides back, and this is where physiology becomes really challenging, is to connect these little dots. So let me connect some of those dots for you right now so that you can go home and think about it. What did I tell you about the size principle? Which types of motor units get activated first? The smallest ones. So what motor neurons get activated first then? So the slow oxidative types get activated first, the small diameter ones. They're the ones that are involved in generating little tension good for postural support, overall can last a very long time before they fatigue. So make sure that you're trying to connect the dots in terms of the physiology of what these different muscle fibers do. And this again is sort of encapsulated in this size principle. One of the reasons why we look at these different fibers and the small diameter axons in these motor neurons, these are the easiest to actually get activated. So they're the ones that get excited first by summated EPSPs. It's harder to do that. It's harder to cause the large diameter motor neurons to become excited or to become activated. And so the slow oxidative ones are recruited by relatively low levels of activity. So as you're doing that initial stretch reflex, for example, the slow ones get activated first because they're the easiest to excite, and they use those small diameter motor neurons. The higher end ones, the fast glycolytic, you need a lot of activity. In order for you to recruit as much muscle mass as you can, you need to recruit and activate the motor neurons that are going to contract all of the fast glycolytic. It's much harder to do that, and this is related to the size principle and activation of these different diameter motor neurons. So we're going to see tetanic contraction a little bit later on, smooth contraction, smooth muscle contraction. And what I'm going to be telling you, again, to connect that dot with what I'm going to be telling you later on, so all of these things are related. In order for you to get the fast glycolytic to become activated, you need your motor neurons to be firing at the highest frequency possible. And the highest frequencies are the ones that are associated with the fast glycolytic. And again, we'll go back and talk about this a little bit later on. Okay, so slow motor units recruited first before the fast ones because they're the easiest. Any questions about the size principle? So small diameter, slow oxidative, easiest to excite. Large diameter motor neurons, always innervating and activating the fast glycolytic, which gives you the most power, but it also fatigues the easiest, and it's the hardest to excite. So keep all of those points clear in terms of the motor unit and the muscle fibers. Any questions on that before we go on? Or any general questions? Okay. So we're going to be looking at how does the muscle actually contract. In the next few slides, we're going to be looking at some of the mechanisms for muscle contraction. So it's not like a typical synapse with the presynaptic element, postsynaptic element. It's not even like the en passant type of synapses. The synapses are really almost like your arm coming down, the axon, and instead of making one or two, it will actually spread out and make multiple connections. It's like a family of synapses that are making contacts with the muscle fiber, or in parallel if you want to think of it. The neurotransmitter that's going to be released from the motor neuron, so here's the motor neuron axon, whether it's small diameter, slow oxidative, or large diameter, fast glycolytic. Once the action potential gets down here, the neurotransmitter that gets released is always going to be acetylcholine, and acetylcholine will then act on what's known as the muscle cell membrane that we're going to call the sarcolemma. And this sarcolemma has embedded on it these nicotinic receptors that respond to acetylcholine. And this structure here that you see is known as an end plate. So it's a axon plus a muscle fiber. We call that a motor end plate for the motor neuron. And it causes this membrane to depolarize. Okay, just like we've seen with neurons, this is also going to depolarize. So again, just to reiterate a couple of points, this is the acetylcholine that's going to be released. Just like in the nervous system, there has to be a mechanism to actually prevent acetylcholine from hanging around for too long, and this is through the acetylcholinesterase enzyme. It's not shown well here on this particular diagram, but acetylcholinesterase is usually found in 3 different places. So it's found deep within the folds of the end plates down here. It's found extracellularly out in the synaptic cleft, and it's also found associated with the sarcolemma. So we find it all over the place. It's important that acetylcholinesterase breaks down the neurotransmitter so that once the transmitter is released, the signal doesn't persist. So again, it will diffuse to the acetylcholine receptor. This is all nicotinic. and we'll go through the nicotinic receptor one more time, and then it will cause its activity to occur. A couple of other things to make note of just for the time being. Do you guys all have taken a biology course like Bio 230 before? Yes? So do you know anything about these other molecules like musk that you see up here? Musk? What do you think, based on this diagram, musk might be able to do? What might its function be? Can you guess or make an educated guess as to what it might be doing? Yes.
Prevent the receptor from accepting the enzyme, the transmitter. So it inhibits this action?
It inhibits the action? Possibly. It is something that I'm looking at in terms of spatial location. So it's right beside the acetylcholine receptor, as your colleague pointed out. What do you think it might be doing? Did you guys talk about that with Dr. Key at all? Like these receptor complexes, they're actually free to float around in the membrane. How do you make sure they're not just wandering around? And it's a principle in neuroscience that we have these clustering molecules that prevent receptors from wandering around. And so for the acetylcholine receptor, this musk molecule actually anchors them at this particular place so that they're not kind of wandering off. And we can use musk to identify the motor end plate. So we can stain for this. We know that we are looking at the motor end plate near acetylcholine receptor types. Okay, so the blood-brain barrier is something that we talked about earlier and something that you talked about with Dr. Key. Peripheral nerves, as we mentioned earlier, don't have the benefit of this blood-brain barrier. We don't have the bony covering. We don't have any of the special astrocytic end feet, nor do we have special capillaries. There's nothing to protect the nerve from the muscle. It's fully accessible by different types of toxins. So this means that different types of toxins that we find environmentally can actually block this neuromuscular junction, that motor end plate that I just showed you, and we can actually classify these different toxins into 3 different types. We can directly block the nicotinic acetylcholine receptor. So one of the different drugs that was found in South America, curare, is a poison that actually will block the nicotinic acetylcholine receptor. So what happens is when a nerve fires and it releases acetylcholine, if curare is there in the neuromuscular junction, acetylcholine can't bind. And because it can't bind, it won't open the cation channels that are associated with the nicotinic receptor, which means that the end plate can't depolarize. You don't get depolarization. If you don't get depolarization, you won't get muscle contraction. And so what curare is doing is to prevent your muscles from being able to contract. Why do you think it's actually a poison? So why is a muscle relaxant bad for you? Because it is. It's really bad for you. We use it in surgery, for example. It prevents muscles from contracting, and it's a good muscle relaxant so that during a surgical procedure, there are no reflexive muscle contractions, etc. But why would we really worry about having too much muscle relaxation? Yes. Could stop the heart. Possibly, I guess. What other muscle groups do you need working all the time, though? Your lungs, yes, but not the lungs themselves, right? The muscles that actually control like how your lungs are inflating, etc. The intercostal muscles in your ribs. So if we have too much of these muscle relaxants, it can actually cause respiratory failure because the muscles in your chest aren't working anymore. And this is one of the reasons why these toxins are incredibly dangerous. There is another type of toxin, a second category known as the botulinum toxin. This is normally found where you have food that is inappropriately processed. Bacteria gets in, and the spores that they release and the bacterial spores actually contain the botulinum toxin. And one of the things that it does is to work on the presynaptic snare complex, and it cleaves the snare complexes so that no exocytosis nor vesicle fusion can actually occur. So what happens is, if you have botulinum toxin poisoning, you're no longer able to cause the release, especially at the portion of the neuromuscular junction, because there's no blood-brain barrier, there's no protection. These synaptic sites are very vulnerable to having the vesicles not being able to release. So no acetylcholine release, you can't actually get the nicotinic receptors to respond, and as a result, this is another form of muscle relaxation. So without this depolarization, no action potentials, no muscle fiber contraction, which means that with botulinum, just like with curare through a different mechanism, the muscle relaxes. Where else have you heard of botulinum toxin? Yes. So Botox. So where do you use Botox? On your face. Okay. Why do you use Botox on your face? Yeah, so the wrinkles that you have, in your skin are actually because the muscles in your face are actually contracting underneath. And by injecting Botox, it will actually cause the muscles in your face to relax, the wrinkles disappear, or they smooth out a little bit. Has anyone heard about what happens with Botox? Like, do you get it once and you never need it again, or what sorts of things do you know about Botox? Yes. Yeah, if you have overuse of Botox and you get lots of Botox injections over a short period of time, yes, your face looks very mask-like. It gets very frozen. But do you have to get it multiple times, or can you use Botox once and all your wrinkles disappear forever? So again, within about a half a year to about a year, the Botox eventually wears out. It gets diffuses away from the site. It gets taken up into lysosomes, etc. It gets degraded, and eventually you need another shot of Botox as the wrinkles start reappearing because the muscles are permanently in a state of excitation. The last type are these acetylcholinesterases, and if you inhibit these acetylcholinesterases, this is the third mechanism. So a lot of different nerve gases and pesticides that contain organophosphates will actually poison the acetylcholinesterase. So remember, acetylcholinesterase, its job, as I just showed you, is to break down acetylcholine at the neuromuscular junction as it's being released, and we don't want too much depolarization to be occurring. So acetylcholine in this case is not being broken down. Acetylcholine stays on the receptor. It keeps activating the receptor, and it continues to cause depolarization of the motor end plate. So the muscles keep contracting, in other words, when we inhibit acetylcholinesterase. Eventually, it will cause paralysis by what's known as depolarization block. And these sodium channels in the vicinity, because there's so much depolarization, the sodium channels in that particular area will be kept in the inactivated state, so you can't actually go back and reset things. So 3 different mechanisms for how we poison the neuromuscular junction, because it's not protected by the blood-brain barrier, all affecting the physiology of how muscles tend to contract. So again, I'm not going to ask you to know any different numbers. We've already looked at this in a slightly different way. Hopefully it's a review for you that the slow oxidative, low amounts of force being generated on the y-axis, if you take a look at it over here, the twitch tension, you can equate this with force. The sort of intermediate, you can see that it takes not quite as long to develop any force, and it's sort of intermediate. The fast glycolytic, it develops force really quickly on the order of several hundred milliseconds, but then it fatigues really quickly. All of this is about twitch contractions, the speed, as well as how this fatigues over time. So this gets into when you start these individual little twitches. So when I generate one of these little twitches, that doesn't really do much for you in terms of muscle contraction. You need what is known as tetanic contraction for your muscles to actually functionally do anything. In fast motor units, and we'll consider this sort of the fast motor units where we're generating sufficient amounts of tension, we start to see the beginnings of these little types of contractions when we start having action potentials going down, causing release. Where we have about 3 action potentials per second, we will generate a little twitch. Another 3 action potentials per second, or 3 per second that you see here, another little twitch. So your muscle kind of spasms. That's not very useful for you. You're not going to write great poetry by having your muscle spasm, right? You're not going to be able to say anything with your vocal cords if your vocal cords are spasming and only spasming. So what you need to do is to increase the rate of activation. You need to increase the number of twitches that you get and recruit more and more of these same muscle fibers together. So by the time you reach about 10 action potentials being delivered to this muscle fiber that we're looking at, by the time you reached about 10 action potentials per second, you can start to see that the twitches start running together. They start to summate. It's still not perfect. What it would look like is instead of a nice contraction like this, so this would be at the upper end. This is a tetanic stimulation, nice and smooth. What you would actually see with a type of trepe or a not quite fully fused tetanus or incomplete tetanus is your muscle would kind of spasm, it would contract. It won't generate the full amount of force, but you'll actually see it as individual little twitches as your arm kind of goes back together. If we increase the speed at which these action potentials get to the muscle fibers that we're trying to stimulate, those twitches run together. And eventually they are fused together, they summate in such a way that the muscle contraction itself is smooth. And how fast do we need to be able to do this? Well, it turns out that we need action potentials at about 30 action potentials per second from your motor neuron in order to get this nice fused tetanus. If we go beyond this, if we increase the firing rate of the motor neurons, so it's theoretically possible to get this up to like 60 action potentials per second. The only thing that will change, because the muscle fibers are only able to generate a certain amount of force, the only thing that will change is getting up to this level, we'll be able to do it much faster. It won't have a slow slope like you see here. The slope would be up around here and generate exactly the same amount of force. You can't change the amount of force being generated after you've reached the maximum, but you can change how quickly you get up to the maximal rate of force. In real physiological terms, this is what happens. This is known as motor unit discharge. And when it is first recruited, these motor units fire at about 6 to 10 impulses or action potentials per second. It starts the process of this tetanic stimulation. Your contractions aren't smooth, in other words, to begin with. However, we want this maximal motor unit discharge or the maximal motor unit force, and we want that nice smooth tetanus. I just told you on that previous slide that we need to get somewhere between 30 to 40 impulses, 30 to 40 action potentials per second to get that nice smooth tetanus. We can only get these motor units, by the way, to start firing at these high rates for very brief periods of time to increase the speed of contraction. You actually can't continue to do this. You would actually cause your motor neurons to fatigue really, really quickly if you try to have them firing all the time at about 60 impulses per second. And so there's a way for your own motor neurons to inhibit themselves or to keep all of this in check. And this is this specialized cell that is sitting right beside the motor neuron. So you can imagine this motor neuron is attached to a whole bunch of different muscle fibers that is trying to fuse into tetanus. Little twitches that it's trying to generate that eventually will cause smooth muscle contraction or fused tetanus. So this motor neuron is actually connected to muscle fibers through this axon that you see here. And all the muscle fibers that it's connected with, this motor neuron and all the muscle fibers would be that motor unit. It starts firing faster and faster, 6 to 10 impulses to start the process of tetanic twitches, but we want nice fused tetanus, so it starts increasing this to a faster rate. So it goes like to 30, to 40, and 50 in order to try to recruit those muscle fibers that it's connected with as quickly as it can. But because it needs to be able to control this, and as a form of control where it's not discharging for too long or too quickly for too long, it has this other collateral. This axon also branches off. So as you start firing action potentials down here, you will also start firing action potentials here, and you will activate at the same time this Renshaw cell. And this Renshaw cell is an inhibitory cell. So as you increase it, you will also start to increase the activity of this Renshaw cell. So at low levels of activity, like 6 to 10 impulses per second, this Renshaw cell really isn't turned on a lot. It's not activated. As you increase it to 30 to 40, it starts to become more and more activated. And once you start reaching 50 to 60, this Renshaw cell now kicks into high gear. And what it does is it will send a collateral back, and it will be inhibitory and turn off the motor neuron. And this inhibitory Renshaw cell is also found in the ventral horn region, and it's there to maintain and to protect the motor neuron from overextending itself and trying to not have too many impulses per second. Any questions about the Renshaw cells or how this sort of regulation works? No? Okay, so I'm going to go through a couple of things in terms of the anatomy of the muscle fiber, and then we're going to come back and talk about the sliding filament theory, one of the best characterized molecular processes that we know in cell biology today, and we have more and more pieces of the puzzle falling in all the time. So we've been looking at acetylcholine. We've looked at some toxins that will affect acetylcholine binding. But I told you that once acetylcholine is released and it binds, it will bind to the nicotinic receptors that are found on that motor end plate or the neuromuscular junction. Now, why is all of that important? So if you were to take a look, this is what is known as a sarcomere, and this unit here is a sarcomere. It is surrounded by that sarcolemma. This whole thing is surrounded by the muscle cell membrane, the sarcolemma, and we've just depolarized the sarcolemma. So the activation of the nicotinic receptors by acetylcholine opens up intrinsic sodium-potassium channels. So tell me again a little bit about what happens when nicotine binds, activates this intrinsic sodium-potassium. What does that actually mean? We just read it together. What does it actually mean? Opens up an intrinsic sodium-potassium channel. What do you think will happen? Sodium flows out of the cell? Yes? No. So sodium flows in, and at the same time, potassium will flow out. So again, both things causing depolarization. So they flow along their normal concentration gradients. So now we have depolarization of the membrane in which we find these nicotinic cells and then receptors. And now this triggers an action potential, which is what we're always looking for in the nervous system. But now it's an action potential that's being generated on the membrane of this muscle cell. And this muscle cell, the cell membrane, actually has some really unique structures that are associated with it. The cell membrane of the muscle cell that we're looking at actually sends projections of the membrane all the way down into the structure through what is known as a T-tubule. So this is part of the membrane. The action potential can actually follow along this cell membrane known as a T-tubule. And one of the reasons why this is important is because as the cell membrane is depolarizing, and as it sends action potentials down these T-tubules, sitting right beside these T-tubules are these structures known as the sarcoplasmic reticulum. So the sarcoplasmic reticulum in striated muscle is the storehouse in the muscle cell of calcium. This is where all the calcium is found inside your muscle cell. And what happens is when you depolarize this T-tubule, you also cause the sarcoplasmic reticulum to depolarize, and calcium now leaves the sarcoplasmic reticulum, and it will flood this area of the muscle cell itself, where we will talk about these different structures. Okay, so that's a really quick overview. I'm going to go through that again for you in some more detail. But this is sort of the way in which a nerve impulse will actually begin a muscle contraction. Any questions on this before we go on? Okay, so again, there's a couple of parts of the anatomy that I need to talk to you about, and I'll define the sarcomere in the next slide. And again, we'll talk about the sliding filament theory, which is very true for all the different muscle types, but in particular for striated or skeletal muscle. And we'll talk very briefly about non-skeletal muscle types, and then we'll talk about pacemaker activity and cardiac muscle, and then we'll come back and talk about smooth muscle at the very end. This is the basic defining unit of any skeletal muscle. Any striated muscle, it looks striated under a microscope because of what is known as the sarcomere. The sarcomere defines the striations. It's a repeating pattern. So this pattern that you see here, this one sarcomere, this pattern repeats itself over and over again in the muscle fiber. And that's why it's a skeletal or striated muscle. I just told you about this sarcolemma. The sarcolemma is the membrane of the muscle cell itself. Would be like one muscle cell that's sort of sitting here. And then this would have the nicotinic receptors. It would cause the influx of sodium. It would cause local depolarization. It would then cause action potentials. Because it's still part of the same cell membrane, these action potentials would run down these T tubules, and sitting right beside these T tubules are the sarcoplasmic reticulum, and they are also voltage dependent, so they open up and calcium will then flow out of the sarcoplasmic reticulum into the spaces that you see here. And under the sarcoplasmic reticulum, we see both thick filaments as well as thin filaments. And these filaments, the sliding filament hypothesis or theory, are the ones that are going to be sliding over top of each other when your muscles contract. Okay, any questions on that at all? Okay, so I think right now is a good time to take a break and to come back and go through this in more detail about how we couple the excitation and the contraction together. Ask. I think it was for 11.
lecture. Do you know if there's like streaming it onto here or whether we have to use our laptop? Oh, we can? Oh, okay, cool, thanks.
Okay, folks, if you don't mind. So a couple of things about your endocrine lecture. It's already available for you online. It's not going to be streamed here. You can watch it at home anytime, as long as you type in the password and go to the link that's provided for you. You can watch it whenever you wish. You can do that today, or you can do it whenever you want. That will remain there for you. And what I'm going to talk to you about over the remaining parts of this is the mechanism behind skeletal muscle contraction and how it works, and you should make sure that you know the different molecules that are involved in this process. And then we'll go through some other muscle types, and then we'll go into some regulation, and then we'll kind of wrap things up. So these are the what are known as the cisterna, these large, the larger sort of parts of the sarcoplasmic reticulum that are right beside the T tubules. And the cisterna of these sarcoplasmic reticulum are really important because as the action potential goes into the interior of the muscle fiber, they run down the T tubules, and then they will be able to open up those calcium channels that are part of the sarcoplasmic reticulum. These calcium channels are voltage dependent, and when they're open, calcium actually goes out of the sarcoplasmic reticulum, and they are released into the space in the muscle cell, which is called the sarcoplasm. Normally in cells, we talk about a cell membrane, and we talk about the cytoplasm. In a muscle cell, the cell membrane is known as the sarcolemma, and the cytoplasm of a muscle cell is known as the sarcoplasm. It's really the same idea, cell membrane, and here the cytoplasm. And this release of calcium that is dependent on an action potential is very, very important in the context of this striated muscle. It is the link between knowing when the muscle should be contracting, when the motor neuron says the muscle should be contracting, and when the muscle itself actually begins to do this. So within the sarcomere, this is again, the general structure. This is repeated over and over again throughout striated muscle. This is the basic working unit. This is the unit that is going to be contracting. It's going to get smaller, and as it gets smaller, it is going to generate force. So if we take a look at the basic components of this sarcomere, we will find a couple of things. We will find that there are both thick filaments, which are comprised of mainly myosin, and we have surrounding them these thin filaments that you see here, which are comprised mainly of actin proteins. We will also see that with the myosin, the thick filaments, we will have these units that are coming out, which are known as myosin heads, that can combine with and bind to the actin filaments that you see here. Stuck right in the middle is this creatine phosphate kinase, and we'll talk about creatine and phosphokinases a little bit later on. So this is the overall structure. These are the filaments that are going to be sliding over top of each other, and this is the general unit known as the sarcomere. There would be another sarcomere that's attached to this on here, and there would be another one to the left of this. So again, it's a repeating structure. All of them are working in the same sort of way, and we have this alternating pattern. So a couple of things that I'll show you, and this number here, 36 nanometers, becomes really important when we talk about the regulation of this binding in a few moments. So this is that same structure, that sarcomere. We've just taken a look at these thick and thin filaments. I wanted you to get an idea that it's not this two-dimensional structure. These are embedded within the muscle fibers. They're surrounding these thick and thin filaments are surrounding each other in a three-dimensional array, and oftentimes when you see it in a textbook, you kind of think of it sort of in a one single dimension, and I wanted you to get an idea that it's in multiple dimensions at the same time. So these myofibrils that you have, the more myofibrils that you have that are working in parallel, the more force that you can generate. So bigger muscles, more myofibrils, more force. So again, like strength being bigger muscles. Bigger muscles are caused by having more of these myofibrils around. So again, just to go over this in one more time before we get into the actual sliding filament theory, is that when we take a look at the sarcolemma, which is the cell membrane of the muscle, so this is the muscle itself, many, many different muscle fibers. Then we take a look at an individual muscle fiber. It doesn't matter, by the way, if this muscle fiber is slow oxidative, fast glycolytic, or the intermediate type. They all work the same way. They just have a slight biochemical differences, but the sliding filaments are the same in all of them. And if we were to take a look at one of these myofibrils and to pull them out, we would be able to see this general structure that we're going to call this sarcomere with the thick and thin filaments sort of alternating. So again, what we're looking at is how the action potentials through the T tubules work their way down and cause these filaments to slide over top of each other. And these T tubules become really important in this whole idea. So the sliding filament theory says this, that the shortening of the muscle itself, as you contract your muscle, one of the things that's happening as you contract your muscle is that the filaments that we've just looked at, the sarcomere, the actin and myosin filaments, they are sliding over each other. And you can imagine as they slide over each other, they get shorter, they contract. Now again, imagine this repeated over and over, and all of those different sarcomeres in a muscle fiber are starting to contract together. And the shortening of the muscle fibers actually has some regulation associated with it. Sorry, whose is this? Yours? Okay. So a couple of things that I need you to keep in mind as we go through. There are two different processes that we're going to be looking at. One is a pure chemical reaction that involves myosin binding to actin. It wants to bind to actin. It's the normal state that actin and myosin want to be able to bind together. And that there is a structural change that requires ATP and the conversion of ATP to ADP that allows for the myosin head that you see here to conformationally change shape. So this movement of the myosin head pushes the actin in a certain direction, and this is what causes the overall shortening. All of these myosin heads pushing on the actin will cause the actin to slide over top of the myosin, which is where this cross-bridge formation is really important. And again, this occurs over and over again on the order of nanoseconds to microseconds to less than milliseconds. It's incredibly fast how these cross bridges form, break apart, form again, and pull the muscle as you're contracting a particular muscle group. So again, going through the different steps, and you can tell that it's important for you to know these different steps on the term test coming up. And if you're going through and you're asking, like, why do you have Drake sort of sitting on top of some of those slides, it's a good slide for you to come back and view again a little bit later on. So these slides that we have with muscle contraction, I want you to come back and review these when you have the opportunity. So again, I'm trying to get you to think a little bit about the way in which actin and myosin bind and how they move together. So we talked about acetylcholine being released. They bind to the nicotinic acetylcholine receptors that are found on the sarcolemma. It causes excitation at the level of the sarcolemma by opening up the sodium channels and the potassium channels. Here you only see the sodium. Sodium flows in. It causes local depolarization. Eventually it will cause action potentials to be generated, and these action potentials will then work their way down, because this is just an extension of the cell membrane, the sarcolemma, these T-tubules, which are the extension of the sarcolemma, all the way down into the muscle fiber. As they go down into the muscle fiber, they are going to encounter these the sarcoplasmic reticulum, and the sarcoplasmic reticulum contains voltage-dependent calcium channels, which now, in response to having a voltage change delivered to them by the action potentials, will then open up. Calcium will then come out of the sarcoplasmic reticulum into the level of the from the sarcoplasmic reticulum into the sarcoplasm, and then they will interact with these thick and thin filaments. Okay, so a couple of really important points is that normally at rest, when your muscle is not doing anything, there's very little calcium that is found inside this muscle cell. So calcium is the actual signal that the muscle should be contracting. Okay. So going back and looking at this actin and myosin structure again, there were some things that I actually wasn't able to point out on that earlier diagram. So when we talk about the thick and thin filaments, and we talk about the thick filaments mainly being myosin, and we talk about the thin filaments that surround it being mainly actin, and from the thick filaments we have these myosin heads, and their normal interaction is the myosin heads want to bind to actin. That's like the normal state of affairs. Just like a normal state of affairs will be how the Golden State Warriors are going to whoop the Cleveland Cavaliers in about 6 games, okay? And that's the normal way in which things are supposed to happen. And so how do we keep that from happening all the time? Why don't they just go ahead and bind each other? Because on the actin filaments that we weren't able to see on those earlier diagrams, we actually have 2 different regulatory molecules. If you took a look at the actin filament, and associated with actin, we have another protein that is sort of interwoven with the actin known as tropomyosin. And tropomyosin is sort of blocking the sites at which these actin molecules might normally be binding. And again, I'll show you this in the next diagram. So actin is surrounding myosin. All of these actin and myosin binding sites are being blocked by this molecule tropomyosin. One of the elements of tropomyosin, which is a separate protein, it is known as troponin. And troponin has this unique feature to it that allows it to also to bind calcium. Okay, so we just saw the release of calcium. There's something that can bind calcium, and this is troponin. Calcium does not bind to tropomyosin, by the way. It only binds to troponin. If you're ever asked on a term test question, it is the troponin that is the binding site, not the tropomyosin for the released calcium from the sarcoplasmic reticulum. So it normally wants to bind, it can't. It's because these sites are being blocked by tropomyosin, and tropomyosin contains another protein element known as troponin. Now calcium is released though. So now things are going to be a little bit different. And this is where those numbers that I talked to you about earlier about the 36 nanometers becomes important. The 36 nanometers that you see here on the actin is where we would be able to have these regulatory elements sort of sitting. So at rest, the myosin can't bind to actin because it has this sort of tropomyosin binding on top of it. It's blocking all the sites that you see here in red where actin and myosin normally should be binding. These myosin heads should be binding at these red sites, but they're being blocked by this tropomyosin. Calcium, though, can bind, and when calcium binds, what happens is the troponin makes a conformational change. Because troponin is attached to tropomyosin, the troponin and tropomyosin complex together starts to roll off of these sites, just like rolling out of bed. And as it does so, these sites are now opened up. So the only way that we could open up these sites was by having calcium binding to the troponin. The troponin then rolls over and exposes the myosin binding sites, and now myosin can do what it's normally designed to be able to do, which is to bind to actin. Are there any questions about this before we continue? So as you're going back and reviewing, make sure you understand what the regulatory proteins are, tropomyosin and troponin. Troponin binds to calcium, and again, the next few steps are ones that you'll also need to be able to understand as well. But any questions on this before we go on? Okay. So this gets into the actual cross-bridge formation. So normally it's being blocked. We can't really get the myosin heads binding appropriately and fully to the actin molecule. And this myosin head actually has ATPase activity associated with it. So it normally will bind ATP and break down ATP into ADP as well as inorganic phosphate. That capacity, though, is being blocked at rest. You have now just signaled through your motor neuron that you would like to contract this muscle, and calcium is now flooded into this compartment. Calcium has bound to troponin. Troponin has then rolled over, carrying with it the tropomyosin that has been blocking this normal binding at 36 nanometer intervals. And as a result, the myosin head can now attach. And because it has ATPase activity associated with it, it will then have a conformational shift. And that conformational shift will cause the myosin head to rotate or move or slide, however you want to think of it. And this power stroke will then cause the myosin and actin to move apart from each other. And it's this ATP binding and hydrolysis in the myosin head that allows this movement to occur. And it will do so, this myosin head can then detach itself in the presence of ATP. So it's just had the power stroke, it has moved the actin, and has moved it far enough that the myosin head is detached from this. New ATP will bind to it. It's now looking for another ATP, another actin binding site that will be coming along. And it may not be the next site that's available to it, because remember, all the other myosin heads are doing the same thing. All the myosin heads are also engaging in this power stroke, so this whole filament is sliding together. However, as soon as another actin site becomes available, it will then bind to it. It has ATPase activity. It will engage in that power stroke again, and the cycle will continue as long as calcium remains bound to troponin, and as long as there is ATP that allows this binding, unbinding, and the movement of this myosin head to be able to continue. Any questions about this before we go on? So you need to be clear on this for your upcoming term tests and potentially for your final exam as well. Any questions at all? Okay, so if we're taking a look at what happens in terms of the actual muscle contraction, this is the structure that we've been looking at. From this Z line that you see here, this sort of zigzag pattern, to this Z line, this is what is known as a sarcomere. That's that one unit. Inside the sarcomere, we have these actin filaments in red, and then we have these thicker myosin filaments in sort of the hatched gray that you see here. Histologically, when we look under the microscope, we would see that the myosin makes up what is known as the A band. It's just a name that we see under a microscope, this dark, thick band. We call it the A band, and it represents all of the thick filaments that we see in the middle. We can also identify the sarcomere through this Z disc, and there are some other elements that are also identifiable. And during muscle contraction, these two ends, the ends of the sarcomere, the Z discs, actually move toward each other. Okay, as the filaments slide across each other in the presence of calcium and ATP, they slide toward each other. They are shortening. And I told you that muscles, as they generate force, shorten. Just think of your muscle as it contracts, it usually shortens. And what is happening is all of these Z discs in parallel are moving toward each other. So the A band, though, actually doesn't change in terms of the size. The overall size of the A band stays the same. And that was something that scientists were using as a marker. They wanted to know, like, how does this muscle contraction actually work? Are we somehow changing the number of myosin molecules? If we were, the change in the A band would be something that we could see. But it turns out that it's no change in the A band. It's just a shortening of the sarcomere as these two Z lines move closer together. So this Z line moves inward, this Z line moves inward, and you can see that compared to the Z lines up at the top, the Z lines are much closer together than they were before. There's been no change in the size of the A band, which represents the overall size of the myosin filaments. And again, the H zone that you see, the space that is normally in between where the actin and myosin are sitting, this zone in the middle, the H band, also shrinks inside, or H zone shrinks in size as well. So some things change and other things do not. Okay, and the A band myosin, that's something that you should make sure you know. The Z discs defining the outer limits of the sarcomere shortening, that's something that you should know. And this space represented by the H zone in the middle, where you have some of the actin and myosin filaments at rest, shortening and becoming smaller, is something that you should know as well. So does the sarcomere change length? Yes, it does. It becomes shorter. And does the H zone change its size? Yes, it does, as the actin and myosin slide over top of each other, but the A band stays exactly the same. I won't ask you, by the way, because I didn't cover it, the I band, which is sort of the region that you have out here where we don't have any of the myosin filaments. So the myosin molecules overall are kept well supplied with ATP. Again, you have to think a little bit about this. I told you that sliding filament model works for all the three muscle fiber types that we looked at: slow oxidative, fast glycolytic, the intermediate type. But it really depends on the amount of ATP. Some muscle fibers will be able to generate lots of ATP for long periods of time, and some will not. So again, a couple of things related to this. As long as there's enough ATP, your muscles will be able to continue to contract. There is another way in which we can actually generate ATP and this is through the creatine phosphate. And this literally is where, at rest, if you have excess creatine, the molecules from ATP can be transferred over to creatine in terms of the phosphate molecule, and this involves the creatine phosphokinase that we saw right in the middle of where the A band was sitting, this sort of M line that you see here. And again, you don't have to worry about M line. That's where we saw previously on another diagram where this enzyme creatine phosphokinase is actually sitting. So this is the way in which ATP is useful for a muscle. We need it to be able to cause detachment, the power shift, the power stroke in the myosin heads, and we need ATP for muscle movement, which gets into muscle fatigue. Muscle fatigue isn't necessarily the running out of ATP. So I just told you that some muscles are really good at producing ATP, some are not. And if you're thinking about it, the slow oxidative type, actually, it doesn't fatigue very easily. How does it actually fatigue over the long term? The fatigue is usually caused first by the uncoupling between the T-tubule depolarization and the opening of calcium. So over time, the voltage-dependent calcium channels no longer respond when there's continual depolarization. And there's that uncoupling. It's not every action potential will open up the sarcoplasmic reticulum. And some of this is also due to acidity. As you are using your muscles, and in particular certain types of muscles, some of the muscle fibers, as they are going through metabolism, they will start to produce more and more lactic acid. And this is the end product of glycolysis, anaerobic respiration, as well as phosphate. So as you are either using up ATP or using up the glycolytic end products, you are producing the end products or the metabolites that will eventually lead to the fatigue within the muscle. So this usually precedes the loss of ATP in the muscle. This is why we say that it's usually not due to ATP. These processes occur before we run out of ATP in the long run. So we talked about how this all occurs, but what actually stops the muscle from contracting? When the motor unit impulses from the motor neurons stop, when there's no more action potentials coming down, there's no more depolarization of the sarcolemma, no more calcium gets released from the sarcoplasmic reticulum that's coupled to it, calcium is actively pumped out of the sarcoplasm back into the sarcoplasmic reticulum. So there's a quick removal, in other words, of this. When you hear about this type of phenomenon, do you think that this requires ATP or not? So it's pumped back in. Does it sound like an active process or a passive process? So this is an active process, this pumping back into the sarcoplasmic reticulum after the action potentials have stopped, actually also involves ATP, and I'll come back to why that's important in a few moments. So there is no more calcium left. All of it is actively transported back into the sarcoplasmic reticulum. There's nothing left to bind to troponin anymore. No more cross bridge formation can take place because there's no more calcium around. Troponin goes back to where it was. It rolls tropomyosin back where it was as a result, and it covers the actin binding sites. But this leads to a very particular case, which is known as rigor mortis. What is rigor mortis? What happens in rigor mortis? Or what does it describe? A stiff animal? Does it have to be an animal? Stiff muscle. Okay, so this is in a dead individual, for example, or a dead animal. What happens is usually the muscles all contract, and it's a very stiff sort of feel when you are looking at the muscles. They're all sort of in a contracted state, which is unusual. And it actually can occur from anywhere between 30 minutes to several hours after either an animal or an individual has died. They stop breathing without taking in oxygen. You stop the oxidative phosphorylation, you stop the ATP production, and without ATP, you can't actually get rid of calcium back into the sarcoplasmic reticulum. Calcium just stays out there. And so it diffuses out of the sarcoplasmic reticulum. It exposes all of the actin myosin binding sites, and all of your muscles across your entire body actually form cross bridges, which is why you get this rigor mortis. So again, no ATP, lots of calcium diffusion. What else happens in rigor mortis? Does the person stay stiff for after they die for forever? No, they do not. Why don't they? Any ideas? Sorry?
They don't have calcium left.
Nope, calcium just stays there. Stays there for as long as it possibly can. Any other ideas? So if calcium is there, all these actin myosin filaments are forming, all the muscles are contracting, there's no regulation at all. What else might cause rigor mortis to disappear? Because it does. You can actually time forensically the passing of an individual by when rigor mortis sets in, depending on the temperature, the age of the individual, etc., to when rigor mortis disappears and the body goes flaccid again. Any ideas? Yep.
Would it just be like a regular decay process where bacteria starts?
Bacteria, okay, possibly. It's kind of related to it. You're right in one sense that with the loss of ATP, not only do we not have the capacity to pump back into the sarcoplasmic reticulum, we also lose the regulation of other internal organelles, including things like lysosomes. And the lysosomes will break open, they'll no longer be regulated, and they start dissolving the actin myosin cross bridges that are found inside the muscles. So the muscles themselves will actually start to break down over time as the lysosomes start to disintegrate. Okay, so I'm going to talk very quickly, and to be honest, I'm not going to give you any questions related to cardiac muscle outside of the 2 slides, and it's more about the concept on these 2 slides. So I'm not going to be asking you to know too much about it. If you're in PSL301, you'll hear a lot more about how the heart works and the regulation of these pacemakers. In cardiac muscle, so we're now switching gears. We're moving away from the striated muscle, which I've been talking about. We're now talking about a different type of muscle, which is the muscle that you find within the heart. Within the heart, we actually have what are known as these pacemakers. And these pacemakers are actually really important because they're specialized muscle tissue that actually can generate spontaneously electrical activity at the level of what is known as the SA or sinoatrial node that you see here within the right atrium. And these pacemakers actually have a very interesting property. They will have a resting threshold potential, and this threshold potential actually allows it to spontaneously start depolarizing and repolarizing by itself. It doesn't need any type of activity to or nerves to actually cause it to start depolarizing. And the depolarization phase of cells here within the SA node is due to voltage-gated calcium channels. Okay, so calcium again playing a role in cardiac muscle tissue. The hyperpolarization is actually due to the opening of calcium-gated potassium channels. And we can actually make use of this, which is what I really want to get to on the next slide. So this is what the overall SA node would look like. By itself, if you were to isolate this group of muscles in the right atrium, this small group of muscles in the right atrium in a dish would have spontaneously this pacemaker rhythm. It would have action potentials firing at this particular frequency, and it's due to the depolarization is due to voltage dependent calcium, and that causes this depolarization. As more and more calcium comes in, more and more potassium channels get activated, which causes this repolarization that you see on the down side of things. The receptors, though, here within the SA node are metabotropic. Do you remember the parasympathetic nervous system that we talked about for autonomic control in the last lecture? The parasympathetic vagus nerve actually releases it releases this acetylcholine, and then it interacts with SA node receptors that are metabotropic. So what are the metabotropic types of acetylcholine receptors? So these are the muscarinic types. The muscarinic acetylcholine receptors that are found on these muscle cells are all metabotropic. And one of the things that it does is it actually, the effect of parasympathetic stimulation is exactly as you see here. So I'm going to show you this, and I'll show you this again down below here. This is the normal pacemaker activity. This is the normal frequency. When you stimulate the vagus nerve, it causes the release of acetylcholine. And when it releases acetylcholine during this period here, wherever you see the black bar, this is where the vagus nerve has been stimulated and therefore where acetylcholine is being released. What do you see happening to the action potentials in this black bar, where you have vagal nerve stimulation and acetylcholine release? Is there any change in the size of the action potentials? They're bigger or smaller? Like really bigger or really smaller? They stay about the same size, right? But one thing that you can see is that we start slowing down the number of action potentials, which is what the vagus nerve does. The parasympathetic nervous system is where you're resting and digesting. The heart isn't supposed to be beating as quickly. And one of the things that happens is this acetylcholine, by working on the metabotropic receptors on these muscle cells, what it actually does is it will increase the time that it actually takes to start this depolarization step that you see here. So normally, before acetylcholine, it would rise, it would depolarize, it would activate the voltage-dependent calcium channels, and then it would start to repolarize here. And then that whole process would begin again. But what acetylcholine does is this depolarization step, before you open up these voltage-dependent calcium channels, this depolarization step in the presence of acetylcholine actually takes a lot longer. So it takes a lot longer for it to start depolarizing, which is the way in which acetylcholine slows down the heart. And this current that you see here, this slow rising current, is actually the what's known as the hyperpolarizing current. And this hyperpolarizing current is reduced or slowed down with the effect of acetylcholine. So functionally, this means that there's a longer interval between action potentials. The action potentials slowing down means that the heart rate slows down, and that's something that the parasympathetic system is supposed to be doing for you. So again, I'm going to go through this fairly quickly to talk to you about smooth muscle, which is the last muscle fiber type. So the majority of today's lecture was about striated or skeletal muscle, voluntary muscle, however you want to think about it. We just had 2 slides related to the cardiac muscle system, because I'm going to wait for my colleague to talk about that when you talk about the cardiovascular system. And I'm going to mention in passing a couple of really important points about the smooth muscle system over the next 2 to 3 slides. So there are different types of smooth muscle. There are smooth muscles that are known as multi-unit, and the multi-unit types are over here. The characteristic features of these multi-unit smooth muscles is that they do not have gap junctions normally associated with them. They are individually depolarized. All of these smooth muscles that you see here in pink are individual units that act independently of each other. And the reason why they can do that is that they all have their own sort of nervous system input through these axonal varicosities that release neurotransmitters. These typically do not have spontaneous action potentials, and therefore we say that they are non-spiking. We don't see a lot of electrical activity in these multi-unit types. The multi-unit type would be represented best by what's shown here on the right. The single unit type is very different, and I'd like you to make sure that you know the properties associated with these different types of smooth muscle. This is normally found in the gut, and this is why you see the small intestine associated with it. It is a visceral form of the single unit. All of these are joined together by different gap junction proteins that you see here. And so when one part of this, when one cell in this single unit type contracts, all of them will contract along with it. It's like setting off a domino effect via the gap junctions, where one contraction causes all of them to contract together. And it makes sense that you want your entire gut to be able to respond as a single unit, not individual cells kind of working separately. The bladder also works in the same way, as well as the uterus. All of these are smooth muscles that act as a single unit, all defined by these gap junctions. And all of these can also generate action potentials as well. So very, very different types. They're all smooth muscle. Smooth muscle themselves are all capable of contracting. So again, some characteristic features of these smooth muscle cells. And again, not to confuse you, but smooth muscle also has actin and it also has myosin. We just don't have them in ordered arrays. We don't have sarcomeres, but we still have actin and we still have myosin. And you can see that instead of forming these sort of parallel structures repeated over and over again, the actin and myosin filaments that you see here, they seem to be more like a network or a mesh within this particular smooth muscle cell that you see up here at the top. And so this makes this very mechanically versatile. It can contract in many, many different configurations. The overall change in length can also be much, much greater because it can contract in multiple directions if you want to think about it. So all of these things can contract in the same way, or they can contract in different ways. And these actin and myosin filaments are in this crisscross pattern. So the length tension, the overall force that's generated, because they're not all going in this one direction, they could be off on 90-degree angles to each other. The overall tension that's generated by smooth muscle tends to be fairly weak. These cells don't have things like troponin. They have myosin and they have actin, but there's no troponin within these cells. What happens is that in this particular case, myosin still needs to form a cross bridge, but it needs a little bit of help in these cells. And one of the things that happens is that with calcium, calcium will activate an enzyme known as caldesmin, and caldesmin in smooth muscles will actually phosphorylate myosin in order to help with that cross bridge formation. Again, with these gap junctions, depolarization goes from one cell into the other. And if we look at the mechanism by which all of this occurs, the entry of calcium here in smooth muscle is from the extracellular space. There is no sarcoplasmic reticulum. There is no internal store of calcium. It has to come from the outside environment. So calcium coming in from the outside environment enables this cross-bridge formation by activating the myosin kinase caldesmin. And this caldesmin will then activate and cause actin and myosin cross-bridge formation. And I talked to you about this metabolic oscillator in the next slide, and these things go through cyclical patterns of excitation, etc. And the major contractions that we see are actually triggered by action potentials, which I'll show you in the next slide as well. Okay, this gets into these metabolic oscillators. There's a lot on this slide, and to be honest, you're most likely going to be seeing a question or two from this slide and the slide that comes after it on your upcoming term test, and likely on your final exam as well. So hopefully you'll be able to take in most of the information that's on here. So I told you on the previous slide that these smooth muscle cells actually are capable of this metabolic oscillation. And here's a good clue for you. This metabolic oscillation involves a lot of different things. It involves calcium influx, and it involves a couple of different enzymes that are associated with this. So these unitary smooth muscles are the ones that have this type of activity. We do not see this in multi-unit. They don't have action potentials. They don't have oscillations. We only see this in smooth muscle. We would only see this in the gut. We would only see this in the urinary bladder. We would only see this in the uterus. And these types of tissues that have these unitary smooth muscles have these spontaneous oscillations of about 12 to 15 cycles per minute, especially in the gut, and you'll learn about that when you talk about the digestive system later on in the course. And this initial depolarization that you see here is due to calcium influx. Now again, this becomes sort of counterintuitive, but the cell actually has this inverse relationship. The unitary cells, the more cyclic AMP there is, the less calcium there is, okay? And then calcium, when we have too much of it, it actually works in the opposite direction with cyclic AMP. So during this time when there's no depolarization, this smooth muscle cell, this unitary smooth muscle cell, will start to build up intracellular stores of cyclic AMP. Cyclic AMP will then bind to and activate a cyclic AMP gated calcium channel, and this calcium channel opens up and calcium starts to flow into this smooth muscle cell from from the extracellular environment. This starts this process of depolarization, and this depolarization in turn will then activate voltage-dependent calcium channels that you see here that cause this plateau. During this plateau phase, where we have lots and lots of calcium coming in, during this plateau phase, the high level of calcium that is now inside the smooth muscle cell will actually inhibit adenylate cyclase. So adenylate cyclase shuts off. That means that cyclic AMP is no longer being formed. And at the same time, phosphodiesterase, which is inside the smooth muscle cell, also starts to break down phosphodiesterase. So phosphodiesterase and adenylate cyclase activity together cause a reduction in cyclic AMP, and over time these calcium channels close and we go back to these resting levels. And now this whole process begins again. Cyclic AMP levels go back up, adenylate cyclase activity goes up, phosphodiesterase activity shuts off, and this whole process begins again over and over, and this is that metabolic oscillator. At rest, this happens every about 12 to 15 times per minute within your gut as well as in the uterus. Do you have any questions about this before we get on to the next slide? So do you know the different mechanisms that cause like the upper plateau phase and the normal resting phase? Hopefully you'll know where cyclic AMP is high, where phosphodiesterase becomes active, where adenylate cyclase is inhibited, etc. So make sure that all the details on here are clear for you. And this gets into the last slide that I have for you, and this gets into a mechanism. So again, I just told you that this is all related to the gut. The gut is a unitary type of muscle. We saw that with the cardiac muscle, that the parasympathetic nervous system can influence the activity of cardiac muscle very briefly through reducing the IH current. And in addition, when we look at the parasympathetic and sympathetic regulation of the smooth muscle activity here, this is sort of the normal activity. The unitary muscles undergo this cyclical metabolic oscillation that you see here, buildup of cyclic AMP, opening up calcium, there's this plateau, more calcium turns off adenylate cyclase, phosphodiesterases also come into play, calcium levels start to drop, it starts to repolarize, and then this whole process begins again over and over. This is also accompanied by the smooth muscle cells actually contracting in this process. But one of the things that happens is that smooth muscle can get regulated by neurotransmitters from either parasympathetic axons like acetylcholine release. And so one of the things that it does is at low levels, this is what acetylcholine does for it all the time. Noradrenaline, though, when it comes in and activates the same smooth muscle, noradrenaline that gets released from sympathetic axons actually inhibits the release of acetylcholine. And what functionally this means is that when you have noradrenaline around, there is no tonic release of acetylcholine. It's presynaptically inhibiting acetylcholine from being released. No acetylcholine, meaning that you don't get this spontaneous rhythmic activity from acetylcholine. And as a result, it actually hyperpolarizes the entire smooth muscle that you see here. So it actually drops down, it becomes harder for it to excite, and it means that it becomes harder for the smooth muscle to contract. So I want you to make sure that you know that noradrenaline presynaptically inhibits the release of acetylcholine, and acetylcholine is tonically active within these unitary smooth muscles, especially within the gut. Any questions on that at all? Okay, so I'm hoping that some of you will tomorrow be able to come by the review. We're going to try our best to stream it. If you can't make it down, again, no worries. I'm sure someone will record it and upload the recording. If you happen to have a question while we're streaming, or there's something that's not covered, I think someone will be checking out the Facebook study group, and they'll relay to me any questions that you have, as well as you can ask questions directly on the YouTube link. I'm going to wish you the best of luck on this. You guys have run, this is actually more like a marathon in this course. At the end of this course, when you sit there in front of this stack of notes, which will be about this high, you'll know that you've actually run a marathon, and you've run a brilliant marathon. Don't worry about the next term test. It'll be about the same level of difficulty. I was asked to make it slightly harder, but I decided that that wouldn't be a really good thing to do. So good luck. I'll see some of you tomorrow, and good luck on the term test.